Hello, my name is Soni Savaipulam Seti. I'm working as a group leader at the Max Planck Institute, but now I'm in Germany. And today I would like to talk about the role of microRNAs in the pathogenesis of pulmonary hypertension, whether they can be a potential therapeutic targets. The first, I would like to give a brief overview about RNAs in general. The RNAs is the one which has ubiquitously expressed the global molecules which are regulating the coding, the decoding and the regulation of several cellular processes, and they are typically having the ribonucleic acids, and they are single-stranded molecules, and there are several types of RNA. The separation is based on whether they code or non-code the protein. So the first one is the coding, the messenger RNA, so which is the one which codes for the protein, and this is also typically called for mRNA. And the second category is the ones which are not coding for the proteins. So in this there come several of them like ribosomal RNA, the transfer RNA, the small nuclear RNA, short interfering RNA and microRNAs. So today my talk will be focused on the microRNAs. And among these non-coding RNAs, why they were interested, as you can see here, in the humans, the non-coding RNAs seems to have the number of non-coding RNAs are tremendously, and they're found with the bioinformatics several thousands of them. As you can see, compared to the protein coding RNAs in the humans, which is only 2%, 98% is the non-coding RNAs. And these non-coding RNAs, they are not only high, but they express the gene expression. And thus they can have a possible role in the development, in cancer and several disease conditions, as well as in the host pathogen interactions. So coming to the microRNAs, what are they? They are the small endogenous non-coding single-stranded RNAs which are 20 to 22 nucleotides long, and they repress the activity of the complementary mRNAs, and they regulate 30% of the mammalian gene products. And the most important and the crucial point of the microRNAs is they regulate hundreds of the mRNAs. That's why you can change a phenotype by modulating a single microRNA. So they have been described very well in the invertebrates and the vertebrates, and many of the microRNAs are conserved from the worm to the humans. So next I would like to say how the microRNA is genome organized. They constitute 1 to 3% of the genome. Some of the microRNAs have their independent promoters, as you can see here, and so they are regulated by the different transcription factors. And the second category is the microRNA coding genomic sequences are present in the intronic region, and they always have the sense orientation, and they are involved in the transcribed with the gene. Or the microRNAs can be embedded or overlapped with the exons of the known transcript and always in the same orientation. How the microRNAs are synthesized? So at first the microRNAs is synthesized as by the RNA polymerase II and as a several kilobase pairs. But later they are cleaved as the primary microRNA, and they are cleaved in the nucleus by DROSHA to the 70 nucleotide transcript, and this is called the pre-microRNA. And this pre-microRNA is transported into the cytoplasm by exportin 5 through the nuclear pores. It's cleaved by the dicer enzymes into 19 to 22 nucleotide transcripts, and it forms a complex with RISC, and then this directs them to the 3' UTR region of the mRNA. So what it does after going to the mRNA, it works on two different modes of action. One is the translational repression. So that means the microRNA binds to the mRNA target with imperfect. So there is it does translational repression when it requires only a partial complementary. It can also cause the transcript degradation. That means the microRNA binds to the mRNA targets with the perfect complementary sequences, and that's why it can cause the transcript degradation. So these are the two modes of mechanism, so which is really important. It's not only causes the translational repression, but it also causes the transcript degradation. So what is the nomenclature when you look any papers of the any papers you see about microRNAs? So the microRNAs are given a number that is attached with the prefix mir by the dash of mir 123. The uncapitalized mir is written, this means the pre-microRNA. When it is capitalized, it shows the mature form. The microRNAs have with the similar structures, except one or two nucleotides. They are written as microRNA 1A, for example, and 1B. The same microRNA which is produced by the genes at different locus, and they are called like mir-1-1 or mir-1-2. So it's a different nomenclatures which is of importance when you read. So what about the history, how the microRNAs came out? These are discovered by the Victor Ambros, Rosland and Rhonda Fenbaum in 1993. The first microRNA was discovered in C. elegans as lin4 is the one. And here you can see there are several milestones of the microRNA research where it started from the C. elegans and later discovered in the plants and later in the leukemias, and which leads to later for several of the in vivo and different disease conditions, the relevance of the microRNAs. So if you see in the recent pulmonary circulation papers, it was shown the number of publications when you type the words of microRNA or the mir comes up. So the PubMed from 2001 to where you have least publications, but in 2011 you see a 5,000-fold increase in the publications. So which suggests that the microRNAs and their role in the disease conditions and physiological conditions is tremendously increasing. As I said, the microRNAs can regulate several target genes. So this makes them as and thus they can function as master switches, and they can regulate several cell functions like the developmental timing, the insulin secretion, fat metabolism, stem cell maintenance, cell differentiation, cell proliferation, and apoptosis. More or less every other function you can think of. Additionally, the microRNAs have been found to be altered in various disease conditions. For example, the functions of the microRNAs in the heart hypertrophy is delineated. So the several of the microRNAs were shown to be involved in the normal heart development, as well as in the hypertrophic responses of the microRNAs are involved, like microRNA 29 is involved in the extracellular matrix component. MicroRNA 21 via MAP kinase signaling can regulate the heart fibrosis. And so in that way they can regulate the heart hypertrophy, the pathological and hypertrophy. And coming to the functions of the microRNAs in other diseases is mainly cancer, where several of the microRNAs were known to be regulated and they can cause several functional consequences. So the microRNAs can function both as a tumor suppressors and the oncogenes depending on what are their downstream mRNA inactivation. So you can see this is the microRNAs as the tumor suppressors, so they can regulate the tumor suppressor as well as the oncogenes. What are the other disease conditions? So these are the ones which are listed here. So they are involved in the neurodegenerative diseases, liver fibrosis, obesity, cardiovascular diseases, and diabetes, and mainly coming to the pulmonary hypertension. So what we thought is microRNAs, first before going to pulmonary hypertension, what is their role? I want to introduce two different ways of targeting the microRNAs. One is the antagomeres, and the other one is the microRNA mimics. So what are the antagomeres? These are the chemically engineered oligonucleotides which are used to silence the endogenous microRNA. Thus the synthetic RNAs which are perfectly complementary to the specific microRNA target. What was recently done is there are several modifications are done so that they are resistant to this RNA degradation. Coming to the microRNA mimics, these are the small synthetic double-stranded molecules which mimics exactly like microRNAs. So when you put this microRNA mimics, it mimics the endogenous microRNA and binds specifically to its target gene and produces a translational repression. So that means you can use it as a microRNA gain of function, and the antagomeres you can use as microRNA loss of function. So the next issue is like why we think that they are involved in pulmonary hypertension, because we recently, from past 10 years, people start thinking the pulmonary hypertension is not only involved, causes the vasoconstriction, but there is a vascular remodeling, and this vascular remodeling mimics a quasi-neoplastic disease, where you can see the hyperproliferation of the smooth muscle cells, the endothelial cells, as well as the adventitial fibroblast. And what is also known is they have an aberrant pro-proliferative and apoptosis resistance of the cells, which mimics like a tumor phenotype. So we thought, so this is a common feature. So since the microRNAs were shown to be so potentially involved in cancer metastasis development, we asked what is the role of the microRNAs in pulmonary vascular remodeling. So one of the targets we focused is the members of the microRNA 17 to 19 cluster, because recently Carusi et al. Has shown in the paper that the microRNA 17 to 92 clusters were upregulated in different experimental models of conditions. Experimental models of conditions, for example in hypoxia as well as in monocrotaline, you can see microRNA 17, 92 and 17 clusters are regulated. And we also confirmed this in our experimental models and human tissues. And the other microRNA of interest is microRNA 21. And it was shown that the microRNA is involved in the neointima formation and then in dedifferentiation of the vesicular smooth muscle cells. And recently it was shown that the upregulation of microRNA 21 is involved in the cardiac hypertrophy in the failing of the heart. And a publication from Sarkar has shown that in the smooth muscle cells, hypoxia induces microRNA 21, which leads to enhanced proliferation and migration. So since microRNA 17 to 92 cluster and microRNA 21 were shown to be involved in vitro or maybe regulated in the pathogenesis, so we want to see what is the functional consequences of its targeting. So we took the strategy of antagomeres. As I said, this is like a loss of function. So the antagomeres is like single-stranded RNA nucleotides. They are complementary to the specific microRNAs and they are chemically modified. As you can see, there is a phosphorothioate and phosphodiesterase changes for stability, and they are conjugated with the cholesterol, so for the better delivery in vivo. So what we have done is to check in the in vivo effects. We took the hypoxia model of pulmonary hypertension. So we exposed the mice to hypoxia to 10% of O2 for 14 days, and after that we started treating them with the antagomere on every third day with different antagomeres. And after day 27 we measured the hemodynamics by catheterization and by echocardiography. We measured for the changes in the right heart hypertrophy, we did the changes in the vesicular remodeling parameters by histology and by morphometry. What we can see here is the microRNA knockdown in the mouse lungs after antagomere. For example, in microRNA 17 is like compared to the antagomere control treatment, antagomere 17, you can see the suppression and it is pretty selective because microRNA 21 and 92 is not altered. And the same is true for the antagomere 21 and antagomere 92. So the antagomeres are suppressing the microRNA expressions there in vivo. So then when you look at the hemodynamic parameters compared to the hypoxia-induced right ventricular systolic pressure, antagomere 17 and 21 decreases the right ventricular systolic pressure, no big changes except antagomere 92 on systemic arterial pressures. And when we measured by echo the acceleration time, you can see an increase in pulmonary artery acceleration time by antagomere 17 treatment and a reduction of the right heart hypertrophy. Since antagomere 17 causes changes in the hemodynamics, we also looked in the vascular remodeling parameters. What you can see is in much simpler way, in hypoxic conditions, you see a decrease in non-muscularized arteries and an increase in partially muscularized and non-muscularized arteries. But when you treat with them with the antagomere 17, you see a reversal. So you see a fully muscularized arteries are reduced and you see also a non-muscularized arteries are increased in this context. So these are the representative pictures how it looks like. So as you can see, compared to antagomere control treatment, antagomere 17, they have a reduced smooth muscle cells. Since it has shown effects in the hypoxia model, we went to another severe model of pulmonary hypertension, that is the monocrotaline model. So what you do is like the rats are injected with the monocrotaline, and after 21 days and 22 days, we started therapy giving antagomeres weekly once. And after 35 days, we measure for the hemodynamic changes and the right heart hypertrophy and vascular changes. And this is what the knockdown efficacy as compared to the antagonia control treatment. Antagonia 17, you see 100% knockdown and in the lung, but then we want to see whether it is also reaching to the target of our interest, that is the pulmonary arteries. So as you can see, there is a 50% reduction of your microRNA, and we will see what happens on with the hemodynamics. So compared to the monocrotaline induced right ventricular systolic pressures, antagonia 17 treatment drastically reduced the right ventricular systolic pressures. It doesn't have any influence on systemic arterial pressures, but the pulmonary arterial acceleration time and the cardiac output, which are measured by echo, are increased. And as well as the remodeling changes is also reversed by antagonia 17 treatment. So then we want to check what are the target genes which are regulated, which are causing the reversal of the vascular remodeling in this context. From the predicted microRNA 17 or another homologous of this, that's microRNA 20, there are several target genes are known from the literature or from the predicted, that is HIF1 for hypoxia-inducible factor, STAT3, IRF1, cyclin D1, P21, and TGF-beta receptor 2, BMPR, and VGFA. So when we try to look for all these changes which are regulated by this, we did the screening for all of them, and we found the important ones which are changed is, we observed a twofold increase in the BMPR2 expression. We observed also an increase in ID1 and no big changes in the TGF-beta receptor 2. And another important target that came up was the cell cycle inhibitor, that is P21. So P21 expression is increased in these in vivo lungs, and also at the mRNA as well as at the protein compared to the controls. And when we did the staining, so since the cell cycle inhibitor is increased, so we checked like for the proliferation effects, what we can see in the monocrotaline and the anti-controls, you see a higher proliferation and higher PCNA positive cells, but with the antagonia 17 treatment, the number of PCNA positive cells are reduced, suggesting there is a reduction in proliferation. So we went in the opposite way. I told you that the microRNA mimetics are the pre-17 microRNA mimics is like a gain of function. So what we done is already loss of function. So we want to see the effects where they are involved in the pathogenesis or promoting the pathogenesis of pulmonary hypertension. So first we checked it in the cells. So we overexpressed the microRNA, which is the precursor 17. So what you can see, this is the overexpression. So you see several folders of increase. But in the same setting, so we can see a downregulation of P21, which is exactly opposite to the antagonia treatment. And most importantly, this pre-17, the treatment increases the basal proliferation as well as the PDGF-induced proliferation, suggesting they are involved in the proliferation of the smooth muscle cells. In summary, I would like to say that when you inhibit the microRNA 17 by using antagonia 17, so you increase the P21 expression, you also increase the BMPR2 and ID1 expression, which causes the inhibition of proliferation. So that means there is a decrease, and further there is a decrease in the remodeling, and which improves the cardiac function. It's apart from our group, studies from our group, we also know from the literature that the microRNAs mimetics or the antagomias can be used in the in vivo, and it can be future as therapeutic strategies. For example, Carbon et al. Shown that microRNA 204 can be possible if microRNA mimetic is regressing pulmonary hypertension, and they applied by the intratracheal nebulization. MicroRNA 20 was applied by intraperitoneally, and we did it in the intravenous. So there is a different ways of route, and then but all of them are mimetics or the antagomias are working in the in vivo conditions. So with this, I would like to thank all the people who are involved in this work, and thank you